Hello, everyone, and welcome to the Interagency Modeling and Analysis Group pre-meeting webinar series for the workshop on integrating machine learning with multiscale modeling. I am pleased to have Dr. John Bachman here from Harvard speaking with us on this webinar. Dr. Bachman is a fellow in the therapeutic science at Harvard Medical School's Laboratory of Systems Pharmacology, and his research focuses on the development of computational tools for understanding the behavior of complex biological systems and the applications of these tools to studying problems of cellular decision-making in health and disease. And John will be talking about the machine-assisted modeling to accelerate biomedical discovery in the INDRA program. So I'll pass it over to you, John. Thanks.
Okay, thank you very much for that introduction. So I'm going to talk about machine-assisted modeling to accelerate biomedical discovery, and everything I'm going to talk about today has been the product of a collaboration with my colleague, Dr. Benjamin Yori, who is also a postdoc at the Laboratory of Systems Pharmacology here at Harvard Medical School, who just unfortunately could not join us for this webinar today. So the problem that we've been trying to tackle is that of generating actionable mechanistic insights from large-scale data in biology. So even as the number of types and amount of data that we can generate experimentally about biological systems has increased, it's often still very difficult to interpret that and to understand why we're seeing the things that we're seeing and to figure out what the next experiment or perturbation should be. And I would argue that this is the result of the longstanding divide between the two approaches to analyzing data in systems biology. And the first is the classic method of trying to understand a system by building a detailed mechanistic model, generally fairly restricted in scope, at modeling the activities of individual genes and proteins and small molecules, and often with a high resolution in terms of time. This could be a model based on ordinary differential equations or even Boolean logic, for example. And then with a model like that, a researcher can ask certain questions by running simulations and then try to understand what kinds of perturbations or what kinds of processes give rise to something like the behavior of a particular signaling pathway here, illustrated with the MAP kinase signaling pathway. On the other hand, we also have the top-down method of analysis, where we take a large-scale data set and we cluster it. We look for enrichment of certain genes or gene ontology terms, for example. And this can point us in the direction of certain correlations and general types of biological processes that might be involved in what we're studying, but often don't give us the resolution, the mechanistic resolution that we might want in terms of thinking about how we might optimize a drug treatment, for example. And one way to look at this is, the bottom-up approach of these detailed molecular models is the MSM part of ML-MSM, the multiscale modeling piece, and then the data-driven inference part on the right-hand side is the ML piece, the machine learning piece. DARPA, the Defense Advanced Research Projects Agency, which has supported some of our work in this area, has actually taken to calling this the first wave of AI or expert systems on the left-hand side and the second wave or statistical inference or machine learning on the right-hand side. And what they've been exploring and what the kind of area where our work fits in is what they're now calling this third wave approach, where we combine multi-scale models and we combine machine learning together in some way to perform more effectively and generate explainable results. In our case, this takes the form of using machines to generate detailed models at a sufficient scale to analyze the kind of large-scale data that we can generate from the lab. So we started on this work with the DARPA Big Mechanism program, which had as its mission to develop technology to read research abstracts and papers to extract pieces of causal mechanisms, assemble these pieces into more complete causal models, and reason over these models to produce explanations. And this was with a focus on cancer signaling pathways. And so the goal here was really to have the machine assemble a large corpus of mechanistic knowledge and use that mechanistic knowledge to analyze data. And we started in this work by trying to conceptually understand what humans do when we build mechanistic models. And the first step is usually gathering information from various sources that could include pathway databases or other curated resources, scientific literature, and also our colleagues who have expert knowledge in this area, and they can sort of summarize it for us. And then that knowledge is aggregated into some kind of representation, and then we filter that knowledge and we sort through it and in different ways. We try to identify where there are errors. We try to understand the redundancies and overlaps between the kinds of pieces of information that we get. Try to figure out which pieces of information are relevant to the problem that we're interested in. We assess different assertions for their believability in different senses, and then we try to infer missing information. And all of that happens in this sort of middle area here, and is an intermediate process. And then once we have a coherent, non-degenerate set of facts relevant to what we're studying, we can generate models. And the models, the types of models, the modeling formalisms that we use will depend on the kind of data that we have and the kinds of questions that we're asking. So that might be an ordinary differential equation model. It's fairly detailed, or it could be sort of a large-scale causal network. And so what we built in this program was a system called the Integrated Network and Dynamical Reasoning Assembler that basically implements this conceptual process of model building from sort of raw information sources like literature. And it has a structure, and this is sort of the structure of how it's used. You have knowledge interfaces to knowledge sources here on the left. They include both curated resources like Pathway Commons databases and other BioPACS resources, multiple different natural language processing, biological event extraction systems, some of which were developed in the Big Mechanism program and others that already existed, and a variety of other formats. So this is not a comprehensive list on the left-hand side. And then INDRA is the assembly engine that takes that information and produces a corpus that can then be translated into different model types. So you get to choose which type of model that you're going to build based on the use case that you have. And just to sort of illustrate what we mean by knowledge assembly, I think a useful metaphor is that of genome assembly, where you have sort of multiple fragmented, overlapping reads or factoids, and you have to assemble them into something that is the best and most coherent representation of the underlying reality. So for example, these are the kinds of extractions that we get from text mining, for example, which is very noisy, still makes lots of errors, even the state-of-the-art systems. And you might take a fact from the MAP kinase cascade, for example, and sort of see it refracted in all these different factoids. So you have information at the level of protein families, so MEK phosphorylates ERK. You have information about individual genes like MEK1 phosphorylates ERK2 at the specific threonine residue. You might have a grounding error or a name entity recognition error where the computer recognized MEK and interpreted as a chemical, methylethyl ketone, and you get an extraction relating that says methylethyl ketone phosphorylates ERK. You get other types of errors like ERK phosphorylates MEK and so on and so forth. And the problem that we have is try to turn this sort of raw material into something that's, you know, most detailed and yet well supported set of information that we can assemble about that mechanism. I'm not going to go into detail of how that works, but that's the kind of conceptual overview of the kind of stuff, the kind of processes that INDRA is implementing on the inside. So this system that basically enables you to take raw material like the scientific literature and databases and turn it into models in a fairly automated, or actually completely automated process, affords us a couple of different applications that I'm going to talk about. So the first is natural language modeling, or the ability of experts to express biological hypotheses for modeling in simple natural language or English, and turn that into executable models. And this is something that we published a few years ago in molecular systems biology. Then we have a larger scale analysis use cases where we try to perform systematic automated explanation of cancer drug effects. I'm going to talk about that one shortly. Then we have built the natural language capabilities into a dialogue system so that a human user can actually interact with the system and ask questions about the model through English. And then finally, in the most recent work in the DARPA Automated Scientific Knowledge Extraction Program, or ASCII program, we've automated this process to build self-updating and self-testing models in different biological domain areas. So first off, just wanted to briefly mention this natural language modeling use case. The idea here is that you can take descriptions of mechanisms, write them down in fairly simple English, Greb2 and Schick, activate RAS, CRF, and SOS, for example, and you press a button to read it with a machine reading system, and then you can automatically get a computable representation of that model. It's represented here graphically, but it actually can be downloaded in different detailed modeling formats, including PySB, SBML, SBGN, VNGL, Kappa, and so on. The idea here is that this type of interface allows modelers to skip the step of expressing things in the formal modeling language and set instead certain policies for how things are assembled. So instead of taking the fact that they want a model that says, MEC phosphorylates ERK, for example, and then deciding how they want to render that as a set of chemical reactions and then writing in those chemical reactions, they simply express it in English and tell INDRA how they would like to have phosphorylation reactions assembled, for example. And all of that happens automatically. And the advantage of this is that it makes the model content much more transparent, reusable, easy for people to understand and modify, and also certainly faster and easier to sort of prototype the models for different purposes. And this is something that is also published from earlier this year. So the second use case is this large-scale analysis of perturbation data. And this is something that we did as part of the DARPA Big Mechanism evaluation. And the idea here is that when you have a data set like this, which is was published by Chris Sanders' lab a few years ago, this is a single melanoma cell line treated with different drug combinations shown on this axis, and then with different proteins and phosphoproteins measured over on this axis. Each of the individual cells in this matrix actually represents a single perturbation experiment with a perturbed node here, or a perturbation, in this case a drug, and then also a particular readout. And often what you want to know, in addition to these large scale patterns, you might actually want to know, well, why is it that when I treat with this particular drug, I see changes in this particular downstream phosphoprotein, for example? And those are the kinds of things that we set as a task for the machine to explain. So in this exercise, we took the largest fold change, largest effects in the data set. These were 50% fold change or greater. And we were able to identify mechanistic paths, detailed mechanistic paths for 20 out of the 22 strongest drug effects. And they're highlighted here. The only ones that that the machine did not produce explanations for were these changes in these proteins downstream of CDK4, for example. So what do I mean when I say mechanistic path? What I mean is, or an example looks something like this. This is a mechanistic path for that would explain how SARC inhibition would cause an increase in the phosphorylation of CHEK2 at this particular phosphosite, for example. And it's represented as a network diagram over here, but also rendered back into English by INDRA in this box on the left-hand side. So you see SARC phosphorylates, this particular tyrosine, or SARC phosphorylated on this tyrosine phosphorylates PAC2 on this particular serine, and that is activating for PAC2, which causes them to phosphorylate RAF1, and so on and so forth. And one thing that's worth noting is that because of the basis of this model in text-mined results as well as database results, and because of the automation of the process, we get full insight into the provenance of each of these interactions. So for example, when I first looked at this explanation, I was sort of surprised that there was this negative regulation between TP53 and PLK1, but I went and I was able to look into it, and then in addition to being able to see that this is actually explicitly encoded in pathway commons, I was able to see a number of sentences from the literature that very clearly illustrate that that this PLK1 downregulation by TP53 has literature support. And if any of these particular sentences were interesting to me, then I could follow up and read these publications, which are also linked to these results. So we are still exploring more applications in this area. One of the things that we're trying to do right now is to identify drug targets from high throughput omics data. The idea here is that a drug has a cascading set of effects that leaves a fingerprint on these downstream observables, and we can actually infer what the sort of root node of those changes is. Another is to explain correlations in gene dependencies. Here, in this case, the cancer dependency map published by the Broad. And we're taking correlations in this data set, for example, this correlation between PTPN11, also known as SHP2, and EXTL3, which is a relatively obscure gene. And the machine can automatically identify that these genes have a relationship based on FGF signaling. And looking further into these edges sort of shows that this is based on the role that EXTL3 plays in the heparin sulfate pathway, for example. So we're looking at doing this systematically across thousands or hundreds of thousands of unexplained gene dependency correlations. So I mentioned as a third use case, a natural language dialogue system. This is something that we've been working on in a different DARPA program called Communicating with Computers. And basically the idea here is to extend the capabilities of beyond of the scientist interacting with a machine assistant beyond simply asserting mechanisms, but being able to build models interactively, query information from the literature, and query the properties of models as they go. And so one use case would be use cases like the analysis of data sets like I talked about before. For example, you could work with the system interactively to understand why a particular inhibitor caused a particular protein to go up or down, but ultimately also in the context of clinical decision-making where the decision-making is based on the activities of particular genes and proteins or mutations. And the way that this works is that a human interacts with the machine, and the machine has certain capabilities that the human doesn't have. That is, they have access to this large corpus of mechanistic information, and then they can run simulations of the model. And we've actually tested this with scientists in the lab. We assign them a certain task, like, for example, how does this particular drug that no one's ever heard of, how could it cause a particular gene like SMRF3 to go down transcriptionally after treatment? And so the scientist works with the machine, makes queries, builds a model, builds up their hypothesis interactively, and then the result is an actual mechanistic model that they can use to explain this finding. And we found that through these human studies, actually, that scientists are faster and better able to do this using this system than just simply using PubMed or Google, for example. So, and finally, what we've been working on most recently is basically deploying this modeling pipeline as an automated process. And this is in the as part of the ASCII program, and the system that we're developing for this program is called the Ecosystem of Machine-Maintained Models with Automated Analysis, or EMMA. And the idea here is that INDRA, basically working with the various machine reading systems that we've been using, monitors the literature as it appears. So new publications are appearing every day, and it scans those publications for mechanistic information and extracts it and assembles it into different models. And models, sometimes, some of these models are oriented around specific signaling pathways or diseases, for example. We're focusing on 6 different cancer types right now. And the key is that we can determine what knowledge is new versus what knowledge has already been incorporated in the model, because we can identify these redundancies and overlaps and evaluate sort of technical reliability of the information that's coming in. But what we've added here is the automated testing and analysis of the new information on the ability of the model to explain high-level observations that are known to be true in this particular area. So I'll talk a little bit what I mean by that. But the result is that we can inform users not only about new facts that are appearing in the literature, but about the impact of those facts on the explanatory power of a model. And in addition, scientists and clinicians can query this system for their own queries that are relevant to what they're studying. For example, they're studying a particular drug or particular target, and they can get notifications that are oriented around that specific sort of causal query that they might have. So it's intended to take a system or a process right now where scientists basically subscribe to PubMed alerts, or they Google and they try to read abstracts and then try to understand what's new, to a process where the machine is really taking on a role of digesting that new knowledge and informing the scientist about new findings that are actually relevant to them based on the interest that they've registered with the system. So it's something that we think of as push science instead of a pull science mode. And we've deployed this system as a web application. So take, for example, a model that we call the RAS machine that models about 226 proteins in the RAS signaling pathway, and extract information about those proteins on a daily basis. This shows the number of statements in the model over time. So you see that new information is appearing over the last several months. And then you can see here that each that the model is subjected to different kinds of tests. For example, that we should observe in this model that AQT1 activates mTOR. We should observe that kinase active BRAF activates MAPK3 and so on. And we actually use the same set of knowledge level information to assemble different types of causal networks at different levels of specificity and granularity. And we see that, for example, for these tests, these are sort of the top tests here, they're satisfied by all representations of this model. So that's all. I'd like to thank my colleague, Dr. Benjamin Guri, the rest of the team that we have working with us on these projects here at HMS, and collaborators including IHMC, University of Arizona, SIFT, people in the Bell Biological Expression Language community, Charlie Hoyt and Dexter Pratt, colleagues at OHSU, and of course support and program management from DARPA. Thanks very much.